Good afternoon and welcome, everybody. My name is Jeffrey Hodge. I'm the Associate Director for the Center for Healthcare Policy and Research. We are joined by the Director, Dr. Courtney Lyles, and we want to welcome you to our seminar series, which also serves as EPI 291. And it's a real pleasure to be able to welcome you today to hear a talk called Summarizing Evidence at the Speed of Policy. And it's about a recent example, and you will be able to participate as well to enjoy the challenges and the interesting things about doing evidence and research at the speed of policy. We have with us Dr. Jeff Tice. He is a professor of medicine at UC San Francisco. He did his residency at UCSF. He did his medical degree at UCSF, and he was Phi Beta Kappa at Dartmouth College. I'm not going to give you the year. I'm not going to give you the year. Dr. Tice's non-clinical work focuses on developing and evaluating risk assessment tools for breast cancer and using those tools to efficiently use screening technologies and preventive therapies to decrease the burden of breast cancer in our community. He also performs technology assessments and support cost-effectiveness modeling and budget impact analyses for new medications, medical devices, and health system interventions for the Institute for Clinical and Economic Review, which we may call ICER. I met Dr. Tice when he was doing an evidence summary for ICER, and I thought, wouldn't it be really interesting for us to learn what you have to do if you want research to be used for a critical decision? If you want to wait until after the decision's made, then you will have all the answers. But if you want to really inform the decision, you may have some difficult challenges to face. And today we have a great example that Dr. Tice will present to us. He has said he welcomes questions during his talk, so feel free to type it into the chat, which I'll be monitoring. Do maybe the actual hand up or the virtual hand up if you would like. Dr. Tice, the floor is yours.
Great. Thank you, Jeffrey. You're way too kind in your introduction. Let me see if I can share my slides. All right, let's do that. And then let's see. Now, are you seeing the full slide or the presenter view?
Perfect. We're seeing perfect full slide.
Excellent. All right, great. The example that I'm going to give is the evidence summary that I presented for ICER at a meeting where we were examining a drug called imetelstat, and it's for anemia in a group of illnesses called the myelodysplastic syndromes, or MDS. And I'll talk about that briefly. And really, I'm happy to be interrupted at any point. I know some people won't be familiar with this disease, and if you have questions about it, I'm happy to try and orient you a little bit more, though I will try and do that through their talk. And the same thing with some of the methodologic pieces. So please feel free to interrupt me. I just want to acknowledge collaborators at ICER. The research lead, Shahryar, is actually on this. I didn't realize he was going to be here today, but I saw him and a few other familiar faces, including some folks I do some breast cancer work with, who were based at UC Davis. So it's nice to see that. But a lot of the work was done by Shahryar and Belen, supporting me in this. The only disclosure I have is that my university has a contract with ICER to do these evidence reviews and support the economic analyses, and I get salary support from that contract. So what are the myelodysplastic syndromes? So it's fundamentally a set of bone marrow disorders where the bone marrow isn't making enough cells, mostly red blood cells. So anemia is a big part of it, and that's what we're going to talk about today, addressing the anemia. But also there can be low white blood cell counts, and so patients are at risk for infections, or low platelet counts, thrombocytopenia, in which case they're at risk for bleeding. And over the long term, patients are at risk for a certain subtype of leukemia called acute myeloid leukemia, or AML. The epidemiology is actually not super well defined for MDS. So the estimates of the number of people living with MDS in the US vary substantially, from 60,000 all the way up to 170,000 people. But we do know it's an expensive disease to manage. with a direct medical cost of over $200,000 a year for many patients. So these patients are sick and require a lot of medical support. The diagnosis typically involves initially, doctors notice, we have a low blood count, cell counts, and we can't figure out why. And eventually it goes to hematology and they do a bone marrow biopsy. And that's really where we make the diagnosis. And there are a lot of different subtypes often that are identified by doing molecular analysis, molecular genetic testing of the abnormal cells in the bone marrow. And that's put together for a scoring, using a scoring system that really identifies the risk for progression to AML. And that's important here because those at very high risk for progression to AML get more aggressive treatments. Those at lower risk, and that's the population we're going to talk about today, are really treated symptomatically and managed until they start to progress and are eligible for more toxic therapies, which hopefully they will never need. But that's why the scoring system is important. It determines eligibility for some of these drugs that we're going to talk about today. The main thing that patients, one of the things that ICER does is we spend a lot of time talking to patients, and we always have at least one patient at our meetings to provide real context for the disease from the patient perspective. But patients really suffer from the fatigue and shortness of breath that's associated with anemia, and it really contributes to poor quality of life. Many of the patients never leave their house because they're so short of breath and just feel tired from this disease. So that fatigue is the number one complaint. There's also a lot of emotional distress because they don't know what's going to happen, what's the prognosis, and it's not a disease, as you probably, I guess that's the most people know about, and so patients don't have a framework for thinking about the disease. And then finally, as I said, it's expensive. Often there are copays. So when talking about some of the medical therapy, one patient in we had focus groups with patients. So one patient said of his copay requirements that we can't afford that. We had actually caregivers, so the patient and this caregiver were there. So financial stresses are a real impact on patients. So what's standard of care? Well, normally blood transfusions when patients are anemic, or ideally, actually, the second bullet, the ESAs or erythropoiesis stimulating agents. That's a fancy term for we're just stimulating the bone marrow to make more red blood cells. And when we can do that, we do that. When that stops working, then we give blood transfusions. But repeated blood transfusions eventually present a couple of problems. One, the body keeps making new antibodies to parts of the red blood cells. So over time, it becomes harder and harder to find units of blood to transfuse. And then red blood cells are filled with iron. And you give a lot of iron, there's too much iron in the body, it gets deposited in the heart, in the pancreas, leading to diabetes, in the heart, leading to heart failure, in the liver, leading to cirrhosis. So we have to remove the iron, which is a burdensome process where you give chemicals that bind the iron and then are excreted from the body, but expensive and not an easy thing to go through. So blood transfusions aren't great. For one subtype, one of the molecular subtypes where we see a deletion of the long arm of chromosome 5, so that's del5q, there's a specific treatment called lenalidomide, which works. And so these patients actually are excluded from the trials I'm going to talk about today, but just in sort of overall context of managing patients with MDS, it's about 10% of patients. And then a comparator that we'll talk about a little bit today is something called luspatercept, and that's another drug for treating anemia, particularly in patients with a subtype of MDS where they have red blood cells with something called a ringed sideroblast phenotype. So this is something that's seen under the microscope. The pathologist defines something as having ringed sideroblasts. So that's the standard care. So for the drug, so we're going to talk about today, the population is those with lower risk of myelodysplastic syndrome, again, lower risk for progression to leukemia, who don't have that del5q mutation, so they aren't eligible for lenalidomide, who are transfusion dependent. And the definition varied in the different trials, but usually it's defined as somewhere between needing 2 to 4 red blood cell transfusions over an 8-week period. And then they had to be receiving their best supportive care and either were ineligible for the ESAs, the red blood cell stimulating agents, or they no longer worked. The ineligibility is because of the endogenous. So we make a normal hormone called erythropoietin that tells the bone marrow to make more red blood cells, release more red blood cells. If that's very high, then these ESAs don't work. Jeffrey, I think you had your hand up.
I do. I want to make sure I understand what's going on. You're looking at a group of people who have a bad disease, a bad condition called MDS, and you're not saying, I want to cure them of MDS. You're saying, when you have MDS, you also often have anemia, and we want to help you with the anemia. Can you go back one slide?
Yep.
And you're saying, oftentimes when you're helping people with anemia, these are the options you can use. And the population you're showing us on slide 7 is a population where the ESA option isn't an option.
Isn't an option anymore, either because they have so much of their own ESA equivalent, our natural ESA equivalent, that giving an ESA doesn't do anything to help, or they've already gotten ESAs, and even with ESAs, they continue to need blood transfusions, at least 2 to 4 over an 8-week period.
To help with the anemia.
To help with the anemia. This is all about anemia. This is not an easy disease to cure. Really, it's bone marrow transplant would be the only cure. And most patients who have MDS are older and have so many other illnesses that they really aren't eligible for bone marrow transplant. So that's why the majority of patients end up in this bucket of mostly supportive therapy.
So if we do a good job, they'll have MDS. And if we do a bad job, they'll have MDS and anemia.
Correct.
Got it. Thanks.
All right, so the main intervention we're going to talk about is imetelstat. So this is a short molecule with multiple nucleotides, so a few nucleotides, an oligonucleotide telomerase inhibitor. So it blocks the gene that extends telomeres, and many people may know these are the sort of the caps at the end of chromosomes. And when they get very short, the cells stop dividing and die. So telomerase keeps these telomeres long, and if you block that, they get shorter, and hopefully the abnormal cells would die. That's the thesis for how this works. The abnormal cells in the bone marrow, they're suppressed with this medicine, so the normal cells can make normal red blood cells and other cell types in the bone marrow, and so patients will no longer be anemic because their own bone marrow is making enough red blood cells. Again, this is studied in transfusion-dependent patients who are transfusion-dependent, but lower risk MDS, and they haven't responded to or are ineligible to the ESAs. And it's an IV drug. It has to be given by an IV infusion every 4 weeks, and it's dosed based on a patient's weight. And the comparators, primary comparison was with placebo. That's the only randomized trial evidence that we have, and really the only evidence that we have for the effectiveness of the therapy. Luspatercept is used in that subset of patients with that ring sideroblast phenotype, and there's a similar trial. And so we're going to do what's called an indirect comparison with luspatercept, because there's a lot of overlap, as I'll show, in the populations who would be eligible for imetelstat and luspatercept. So the key outcomes from a patient perspective, it's quality of life, fatigue, but the FDA guidance is the primary outcome was transfusion independence for at least 8 weeks. So that's an 8-week period without needing a transfusion, and this is in patients who initially were requiring 2 to 4 transfusions over an 8-week period. So that's the primary outcome in the randomized trials, but we like to see improvements in quality of life, fatigue, and then we want to monitor for harms as well, balancing these benefits. So what are the important adverse events?
Dr. Tice, we have a comment. I wondered if you wanted to respond to it. Dr. Rogan in the chat writes that around 2000, Medicare coverage in California restricted the use of erythropoietin drugs for MDS because they were ineffective. Some practitioners, Dr. Rogan continued to prescribe them, so Medicare was compelled to write a local coverage decision to alert practitioners Medicare will refuse to pay. Um, Thoughts about that?
Hi, Dr. Rogan. Nice to see you again. I haven't seen you in years. So I don't know the details. Certainly, the treatment guidelines for MDS and the majority of the hematologists who we spoke with who care for patients with MDS still do use ESAs. Though it sounds like there may be evidence that they're not very effective. I didn't look into the details of that. I just know that that's what the guidelines generally recommend. All right.
That's
I don't have the chat up, so if there's a follow-on question, I'm happy to address it.
super. And just before you continue on, I just want to clarify, you aren't doing this review because you need another publication and it might be it doesn't really matter who sees it and it's kind of going to be buried in the academic literature, right? You're doing this, the evidence you're about to show us, you're reviewing it because it's going to be important, right?
Yeah, I will step back and talk a little bit about ICER. Yeah, I mean, it's actually some of the work that I do that I feel is most impactful, and I feel it's really gratifying to help with this process. ICER times these reviews for when new drugs are about to be approved by the FDA. So we try and have our complete process, including the public meeting and our recommendations at that time. And the real goal is to impact the discussions around price. So part of what we're not talking about today is the economic evaluations, but we look at the value, the added benefits of these new therapies, and we say, how much should we pay for that? And we give some estimate of that based on standards that are used in the United States and around the world. And we try and have that available right at FDA approval, because that's when the pharmaceutical companies choose the price for their drugs and put it out there. And it's also when payers negotiate with pharma about what will actually be paid. Nobody pays the list price. Everybody pays less than the list price, but how much less depends upon your ability to negotiate. And so we try and put evidence out there to help with those negotiation processes so that the prices end up being aligned with the value, the clinical benefits for patients. So sometimes a $2 million therapy, we say, is actually a good value because it improves patients' lives so amazingly. So some gene therapies for things like sickle cell disease or hemophilia, these terrible hereditary diseases, which are really disabling for patients, or hereditary blindness. We actually have said it's reasonable to pay a couple $1 million for that therapy because of the impact it has on patients' lives. On the other hand, sometimes drugs that only cost $10,000 or $15,000 a year, we say, are way overpriced. And it has impact. So sometimes companies have used our approach to actually price their drugs. Another time I did work on a cholesterol-lowering class of drugs called PCSK9 inhibitors. Very, very effective. Priced at around $15,000 a year. We said, yeah, $4,000 or $5,000 a year would be reasonable. Nobody covered. The insurance companies made it very hard to get the drugs at the $15,000 a year price. And within 2 years, the companies dropped their prices to about $4,000 to $5,000 a year. So we are having an impact on drug pricing in this country, which, as we all are aware, is a huge problem. And it's really meaningful work to have some influence on that. I've worked on 2 drugs for the original CMS negotiations, and I'm working on 2 more drugs right now for ICER, where again, we're giving public comment into the process for negotiating the price of these new drugs, where Medicare is actually allowed to negotiate on the price or a subset of drugs. So again, giving input there too. So yeah, that's the sort of context and why it's meaningful. I had done some cost effectiveness analyses early in my career, but felt like, yeah, as you said, it was just going out into the ether and it was academically interesting, but not having much of an impact. But ICER's work, we're really like organizations that most countries have to support their national decisions about covering drugs and what they're going to pay for them. ICER is filling that void in the United States because we don't have that. And we actually make our models available online. So insurers can put in their own costs for hospitalizations and put in their own patient characteristics and rerun our models to more closely align with their actual costs and patient populations. So we try and support making our analyses as relevant for each payer, and the pharmaceutical companies use our models as well. So anyway, little side topic, but that's why this is impactful, and why wrestling with the clinical evidence as it is, this is very different from often what we're doing later on. These are reviews that are done right when something is about to be approved, so there's no real world evidence. So we can't use real world evidence for these newer reviews that I'm doing to support the CMS negotiation process. There, the drugs have been out often for 10 plus years because of the rules in the legislation that allowed for the negotiations. In those cases, we are using a lot of real world evidence as part of the evaluation of the value of the therapies. But for most reviews, and for this one here, there will be it hasn't been FDA approved, so there's no real world evidence. Nobody's using it. The only evidence we have are the early clinical trials. All right. Well, maybe way more than you wanted to hear than about ICER, but clearly I'm passionate about it. I feel like this is really soul satisfying work. So the key trial for imetelstat is called the iMERGE trial. It was a randomized trial, phase 3, 2-to-1 randomization, so twice as many people got the active therapy as a placebo, and everybody was blinded until the end of the trial. The patients had to have lower risk myelodysplastic syndrome by the IPSS rating scale. And their definition of transfusion dependence was at least 4 transfusions over an 8-week period. And the primary outcome was the proportion of patients who achieved at least 8 weeks without a blood transfusion over the 1-year timeframe of the trial. So that will be all of the evidence for the comparison versus standard of care or placebo for one clinical question that I'll ask you later. The second will be a comparison with luspatercept. Again, there's just one randomized trial, a similar design, 2-to-1, blinded. I realize some people don't like double blinded, as patients are blinded, staff is blinded, investigators are blinded, statisticians are blinded throughout the trial period. They used a slightly different version of the IPSS, a revised scale, so a slight refinement, but again, lower risk MDS. And as I'll show you, the two populations in the two trials end up being very similar. Their definition of transfusion dependent wasn't as strict. It was only at least 2 units over an 8-week period. And the one big difference is all of the patients had to have the ring sideroblast phenotype. And their primary outcome was the same, transfusion independence for 8 weeks, but only over a 24-week period, though they do report 48-week data, which is what when we're comparing iMERGE to Medalist, I'll use the 48-week data to make it a little more comparable, though it won't be identical, the outcome. Here are the baseline characteristics of the two trials. You see the ages are very similar, despite slightly different inclusion criteria. 72 in iMERGE, 71 in Medalist. About 38% were women. Hemoglobin levels were comparable. So normal hemoglobin is, for those who aren't familiar, is 13, 14. So these patients have half as many red blood cells as the average person, and hemoglobin below 10 is usually when people get symptomatic. So these patients are very symptomatic. And despite the differences in the requirement for transfusion dependence, being 2 versus 4 over an 8-week period, for the Medalist trial, the mean transfusion burden was 5 transfusions over 8 weeks versus 6 in the iMERGE trial. So even though the inclusion criteria were different, overall they looked similar. And again, for the ring sideroblast phenotype, 2/3 of patients in the iMERGE trial also had the ring sideroblast phenotype. So there was significant overlap in the two populations. So when I'm comparing luspatercept to imetelstat, we'll focus on that subpopulation. So the key outcomes for the iMerge, so this is the imetelstat trial. 40% of patients had at least 8 weeks where they were transfusion independent versus 15% in the placebo group, highly statistically significant. Secondary outcomes, those who became transfusion independent, that lasted on average 52 weeks for those who received imetelstat versus only 13 weeks for those in the placebo group. So clearly a more prolonged transfusion independence translates into much greater improvements in quality of life. Fatigue is measured by a scale called the FACIT fatigue, and half of patients had a significant improvement, at least a 3-point improvement for 2 4-week cycles versus 40% in the placebo group. This probably was not statistically significant. I think we ran the numbers ourselves and it wasn't, but a trend towards improved fatigue. There was really no difference in progression to AML, 2% in the imetelstat group, 3% in the placebo group. Sidebar, there was some hope that imetelstat would be disease modifying, this hopefully killing off some or not allowing the abnormal cells to extend their telomeres and thus prolong the disease or contribute to disease. It was hopeful that this would that imetelstat would change the natural history of the disease. Certainly no evidence to date, though the trial is really underpowered to look at that, but certainly the trend is in the wrong direction here. And then finally, similarly with mortality, a higher percentage of patients died. So you get a sense these patients are older, you saw in their 70s, but also have a lot of comorbidities if 30% of them died in a year, 25 to 30%, so a very sick population. And for the sub set where we're comparing imetelstat to luspatercept, so here we're looking just in patients who have that ring sideroblast phenotype. You see the sample sizes here and the proportion who achieved that 8-week transfusion independence, 45% in both trials for the active treatment, imetelstat and luspatercept. A few more in the placebo group achieved transfusion independence in the iMerge trial versus the Medalist trial, but the results look fairly similar. Let's see, all right, sorry. There we go. We did do a kind of meta-analysis called a network meta-analysis, or an indirect treatment comparison. So the way this works is, we have data comparing imetelstat to placebo, data comparing luspatercept to placebo. The populations are really similar or the same, and the outcome is the same. You should be able to get a sense of, even though we don't have head-to-head data comparing imetelstat to luspatercept, we can look at how they both compared to placebo and estimate how they should compare to each other. And so that's what is done here. The relative risk for imetelstat versus luspatercept for transfusion independence was 0.9, means it did a little bit worse, but remember, it was actually 45% in both groups. The real difference is a little bit of difference in placebo response, but really wide confidence intervals. There's probably no difference, but certainly no evidence that imetelstat is better than luspatercept. And you see the comparisons with placebo both had significant benefits compared to placebo. Luspatercept a little stronger benefit. Limitations of these indirect comparisons. I mean, ideally, we would have all of the data and different outcomes for both groups, particularly for imetelstat. The company didn't provide us all the data we asked for in this subgroup who had the ring sideroblast phenotype. The populations clearly are a little bit different because of the different inclusion criteria. And both trials are pretty small, so as you saw, the confidence intervals are wide. So we don't have a lot of confidence in the NMA, but it gives us some sense of the relative benefits of the two therapies in the subgroup with ring sideroblasts. Harms. So I put both here because you see imetelstat had a lot more discontinuations due to adverse events, 16%, whereas nobody discontinued the placebo due to adverse events in the iMerge trial. Luspatercept trial, the Medalist trial, similar amounts of patients discontinued due to AEs, so probably not a signal for luspatercept causing much in the way of adverse events. There were a lot more grade 3 or 4 adverse events, I'm not going to go through what that all means, but a grade 5 adverse event is death. So grade 4 is really serious, usually involves hospitalization, something that really patients are really sick. And grade 3 and 4 are really pretty serious. So you see almost twice as many grade 3 or 4 adverse events in the imetelstat group versus the placebo group in the iMERGE trial. Similar numbers for luspatercept and placebo in the MEDALIST trial. And what kinds of adverse events are these? They were mostly things that happen with advanced myelodysplastic syndrome. So neutropenia is a low level of the type of white blood cells that mostly fight off bacterial infections. So people with a neutropenia and a fever get admitted to the hospital, put on broad spectrum antibiotics. They're often in the ICU. In this case, they didn't see an excess of that fever plus neutropenia, but you see much more neutropenia in patients with imetelstat versus placebo. They also saw more serious levels of low platelet counts, that's thrombocytopenia, so at risk for bleeding. And despite this being a treatment for anemia, they actually saw more grade 3 or 4 anemia in the imetelstat group versus the placebo group. Whereas you see there were really no differences between luspatercept and placebo in the MEDALIST trial. So you don't have the same degree of adverse events. So controversies and uncertainties, this high incidence of grade 3 and 4 neutropenia and thrombocytopenia. And I didn't present these data, but there was actually a higher incidence of fatigue as an important adverse event in the imetelstat group than in the placebo group, 29% versus 22%, though as I presented before on the scale that measures fatigue, nominally the imetelstat group improved more in terms of fatigue. So a little bit of contradictory information in fatigue as measured by an adverse event versus the scale. We don't really have long-term data, so the question of whether we actually could improve long-term mortality for patients who receive imetelstat. Again, I highlighted we really don't have enough data to confidently compare imetelstat and luspatercept in the ring sideroblast group, though we have some data. So I will say there is substantial unmet need. Talking to patients, they were devastated by the need for going in for transfusions. For many patients, they had to travel long distances to get to transfusion centers where they could do do the testing to find blood units that they're not that they won't destroy with their immune system because of antibodies, and then the time to give a transfusion and stuff. It's really burdensome, and they just felt awful leaving the house. We also look at the impact on caregivers. We didn't have any data on it, but likely long-term transfusion independence would also improve the caregiver's quality of life. So when we're looking at the overall value of these drugs, we try and incorporate other things, not just clinical benefits and cost. So I just thought I'd share some of those things that we look at here. We get a lot of public comments that we respond to. The main one for the clinical evidence was again highlighting the lack of complete data in order to do a really careful comparative effectiveness between imetelstat and luspatercept in the RS positive subgroup. So the summary, compared to best supportive care, so this is not compared to luspatercept, there's clearly a significant reduction in the need for transfusions. But that didn't translate into a significant improvement in fatigue or quality of life. And there were substantially more harms. So these grade 3 and 4 adverse events, including the thrombocytopenia, neutropenia, and anemia. And it's a one small randomized trial. So at best, we have moderate certainty on the improvements in the need for transfusion. So we rate these on a scale that ICER has. In this case, we said it's promising. Definitely some hints of good things, but it's balanced by a lot of hard things, bad things. And so the net benefit we really felt like was inconclusive with the level of data that we had here. For comparing imetelstat to luspatercept, we said it was insufficient. Again, the data we had looked like they had similar impacts on patients with low-risk MDS who had the ring sideroblast phenotype, but was indirect, small trials, and We didn't see the same harms with luspatercept. So it may be actually that luspatercept is a better drug for patients who have the ring sideroblast phenotype. But we had a lot of uncertainty, so I rated the evidence as insufficient. So this gets presented to the panel, and then the panel actually votes both on the clinical evidence and the economic value of the therapies, and we'll come to that. But I think we want to take a little bit of time to talk now. And I don't know, there were a couple of questions that were put out. Dr.
Yeah,
Hodge, do you want to bring those up, or do you want me to put them up, or how would you like
so
to?
before we do the voting, if it's okay to hold off on the poll, is it possible to back down the poll for just a second? So what we're going to do is we're going to ask you to vote as ICER would vote. So basically, they're going to be, if you were on the ICER committee, there would be at least two votes. One vote would be on the clinical evidence, which we've just heard Dr. Tice present, and you'll be given a chance to ask any questions before we ask you to vote. And then you'd have the clinical vote. The second vote, which we aren't going to do today, is about the value. So that's when you've already thought about what you're getting or the clinical stuff. And now we're talking about price. So although we won't be having the clinical vote, Dr. Tice, I'd like you to answer Dr. Rogan's question at the end when
Okay.
you astonish people with your last slide. So we'll come back to value in just a moment. But before we ask the audience to vote on the clinical question, does anyone have any questions? Because if you're on the ICER committee, this isn't happening in some dark room. This is happening on the internet. And if people didn't see it on the internet, they're going to see it on YouTube. So you got patients there, you got providers there, you got the people who make the products there, and you want to answer the question about the evidence. So anyone have any questions about the clinical?
And we have specialists there, so I don't have all of the hard questions about MDS. I get to turf to the specialists in the room. So, but it's a public meeting. I think it's one of the unusual things. So all of the national bodies that do this in England and Canada and France and Germany, around the world, Scandinavia, they all do it behind closed doors. We do this in the public, which is one of the unique things.
So I'm going to start with the questions because if you're going to force me to vote like everyone else, again, this is totally voluntary. You don't have to vote if you don't want to. Also, it's anonymous, so we're not going to call you out and say, Dr. Rogan, how could you vote that way? So I'll give an example. Hey, Dr. Tice, I noticed you found just two trials. One was drug I versus placebo. The other was drug L versus placebo. Why didn't you do a better job finding studies? Because there were only two trials and neither one was a head-to-head. Are you sure you did a good job on your systematic review? You only found two studies.
Yes, I'm confident that we found all the data. So we do a standard systematic review. We put the whole analysis plan out on a public site. But more importantly, we talk to patients, we talk to patient groups, we talk to specialty societies, we talk to clinical experts, and we talk to the companies. The companies always point us to all of the available evidence, including meeting abstracts, and they always provide us with the posters or slide presentations from those meetings. And we also give them an opportunity to provide us with evidence that's academic in confidence, so something that we agree not to reveal for at least a year and a half, though we do put a time frame on it, really giving them time to publish that data. But if they think it's important for us to know for the value proposition or for understanding the clinical benefits, we ask them to provide us with that information. So I'm always confident that we have seen the totality of the evidence, because we really are talking to all players. But most importantly, most of the drug companies are very engaged with us throughout the process and really want to make sure we have all of the evidence.
Super. Are there any final questions for Dr. Tice before we ask you to vote? There'll be two voting questions. One will be comparing drug I to best supportive care or placebo. The other will be comparing drug I to drug L. Remember, there was no drug I versus drug L study.
I will say, in fairness to everybody on the seminar today, we do have the voting members do have access to the report, which Dr. Hutch put in the chat, the link. And there are more outcomes are described, for the presentation and the public discussion. I'm hitting the high points, and sometimes subtle other areas are brought up, and certainly the voting members do have access to, the clinical trial reports and more data that I've written up in the full report, which, including the appendices, is often 300, 400, 500 pages long. So, people are voting on a little bit more than just what I presented here.
Good point. Thank you, Dr. Tice. Bill, are you okay to launch the poll? Again, this is voluntary. It's anonymous. And there you can see it on your screen. The first question says, is the current evidence adequate to demonstrate the net health benefit of drug I plus best supportive care, showing that it's superior to that provided by just best supportive care alone? And the second question is about drug I versus drug L. I don't know. Can you guys see that on your screen?
I can see it on mine.
Cool. Good. We got 4 people who have voted. We have 33 people participating. Let us assume at least 10 are at lunch. No, I'm joking. Everyone's logged in here. So we're at 9. We're at 10, 11, 12. Oh, this is so good. 15. We almost are going to break 50% response rate. 17. We got 17 on question 1. I think we can get to 20. Dr. Tice, what do you think?
I think so. We'll let Jerry vote twice.
I can't vote. I was looking at other stuff. Sorry. I was looking at Medicare policy.
We're at 19 people having voted. We just need one more person. Order now. I can't vote, or I've already voted, so I can't do that. Just one more person. 3, 2, 1. Before I show the results, anyone want to guess? Anyone just take a guess? You think there's more yes, more no? Again, the question is the current evidence adequate to demonstrate the benefit of drug I over best supportive care? And here is what you voted. And I'm going to share the results now. Can everyone see the results?
Yep. So fascinating. So 68% said yes, there was adequate, which so you went with the FDA, who did approve this drug, but the majority said no for the comparison versus luspatercept, the vast majority. All right.
It's okay for me to stop the share?
Yeah, it's good by me.
Okay.
I want to, so let me see if I can advance my slides now again. Let's see. So we did that. Voting questions. Let's see. I'm trying to remember. Let's see if I have my, I can't see my, my recollection, and I'm sorry, I can't see my notes on my slide, but I believe that the ICER panel voted.
I can show the vote if you want me to show the
Okay,
vote.
yeah, I think it was 11-4.
Yeah.
That the evidence was inadequate. They voted no. They did not think there was adequate evidence. And then I think it was 15-0 for no, again, a no vote versus luspatercept is my recollection. So is that correct, Dr. Hutch? Okay.
Great memory. Yes. It's as if you were there.
Those things slip away at my age very quickly. So great. You want me to go to that? That last. So the FDA approved this in June, and that's around the time we had the meeting. The annual cost. So if somebody actually responded and continued to take this for a year, and remember, of those 40% that did achieve that 8-week transfusion independence, the average length of time where they remained transfusion dependent was a year, 52 weeks. So that the charges from the company are $365,000 a year if they continue to get this therapy. So hugely expensive for something that isn't changing the disease course. You know, you're not preventing progression of MDS. You're not preventing death from MDS or other things. You're just preventing transfusions. It's a remarkable amount of money. So as you can imagine, that we did hold votes on value, and the panel consistently said we thought it was low value, that this was not a high value intervention for patients, despite the fact that it probably works, and some patients probably get good benefit from it, but the majority don't. And it's very, very expensive for modest benefits.
So we're going to open the floor. Is that okay, Dr. Tice?
Please,
So while
take
I'm-
down the slides, and should I stop sharing?
Yeah, that'd be great. That'd be great. So while I'm asking for questions, which you can either type into the chat, you can actually raise your hand, turn on the camera and show us your hand, or you can virtually raise your hand. While I'm waiting for your questions, I'm going to ask Dr. Tice to reflect on Dr. Rogan's question. And again, I understand, Dr. Tice, you're not speaking, you're not a spokesperson for ICER, but the question is asking about when ICER finds a drug is too expensive, or let's call it low value. In your opinion, does the finding have any effect on payers' payment amount? And if yes, which ones? And then there's something that follows after.
Well, it's always hard to know, because the actual price paid by payers is always a business secret, and they won't tell us because they want to jeopardize their relationships with pharma. But certainly they use, you know, everybody uses our reports. All important payers use our reports to guide their negotiations. So it certainly has an impact. I gave the example before where clearly we had a big impact for PCSK9 inhibitors, where they were initially priced at $15,000 a year. We said $4,000 to $5,000 a year was reasonable, and within 2 years, the two drugs were then priced at $4,000 to $5,000, or about $5,000 a year, which with rebates, which typically are around 30%, though again, different companies negotiate different rebates. It was in line with what we thought was a reasonable price for the benefits in terms of preventing strokes, heart attacks, and death from cardiovascular disease. So we definitely have an impact. It's not as impactful as what happens in other countries, Canada, England, Germany, France. I mean, we evaluated, for instance, the weight loss drugs that are all the rage, you know, the Ozempics and Wegovys and you know, we said they're overpriced. You know, the companies still charge, you know, $12,000 to $13,000 a year here, where it's $1,000 to $1,500 a year in Canada and England and France. So everywhere else has negotiated a much lower price. So we're having a little bit of an impact, but not as much as we would like.
And then there was also a follow-up here. Again, it's possible you may not know the answer. It says, is there discussion with the administration or Congress about using ICER findings as a basis to reduce Medicare reimbursement for selected drugs to replace current law?
I'm sure there's discussion, and we're hopeful that you know, the small step forward with the IRA legislation that allows for negotiation of drugs that have been out there for, you know, 12 years without a generic competitor and stuff, that those negotiations, ICER is submitting our analyses for a couple of the drugs to illustrate how this sort of analysis could be used for those negotiations, but it certainly could be used at approval of the drugs in the initial negotiations around reimbursement by at least Medicare and Medicaid. It's not routinely done now, but I'm sure it's on the radar screens of you know, some members of Congress. I don't expect to happen anytime soon. And hopefully we'll see some benefits from the initial negotiations, and then maybe there'll be a move to do this earlier in the process of negotiations of drug prices. But yeah, I think it's a long road, but we certainly hope that we're, that's the eventual path that the country will take.
So if you have questions for Dr. Tice, you can actually raise your hand. You can virtually raise your hand. You can type something into the chat. Dr. Tice, as a researcher, and you said this was soul satisfying work, but it feels like there's a lot of pressure. There's a new drug coming out. The patients really want it. The clinicians really want it. The people who make the product really want it. And it seems like not always do you find like really clear, no uncertainty at all kind of results. How do you balance that uncertainty?
Yeah. Oh, absolutely. I also on guideline writing committees where we expect a much higher level of evidence before we're going to make a recommendation at the guideline. So it is a tension, but this is a time when decisions are being made, and so we need to put out the best evidence at that time. If we think there's new evidence, and we think it's really important. Again, the PCSK9 inhibitors are a great example. The initial approval was just on the basis of lowering LDL cholesterol, but the FDA required longer, larger trials with outcomes of stroke and heart attack. And so when those trials were published, we redid our analysis and updated our report to reflect the real clinical outcomes, and reassessed. We actually weren't that far off the first time around, which was reassuring that the modeling that we used on the benefits and harms actually worked. Yeah, so we do that. I'll give another example though of a different thing. One of the things that we get some pushback for is we also look at the potential budget impact, which isn't traditionally done. I did early on some of the new drugs for hepatitis C, which were a slam dunk. Yeah, so much better than prior therapy, so much more effective, better tolerated. I think Dr. Rogan was present for some of those meetings when I presented those evidence. And we said, oh, they were high value. The cost per improvement in life was actually in line with the clinical value. The cost was in line with the clinical value. So we said they were high value, but there were so many people who needed to be treated that this was going to be a huge short-term budget impact. And so we highlighted that and put out a budget impact alert. The VA did nothing about it, and they ran through their entire pharmaceutical budget in a month. So they had no more money for chemotherapy. They had no more money for blood pressure drugs. They had no more money for drugs for diabetes. So they had to go to Congress for special appropriations to give to fund pharmaceuticals for the rest of the year, because they put no brakes on the availability of these new drugs, which we said were high value. But again, there was such demand and such a large population who needed these drugs. So there are all kinds of tensions in the room. And we spend a lot of time, after all the votes, we actually have a policy roundtable where, again, the clinical experts, specialty societies, public and private payers are at the table, patients are at the table, and the companies are at the table. And we say, how do we set things up so that the patients who really need this get this, and that this is done in a thoughtful way. And so there's an interesting policy discussion about how things should happen, even beyond the votes on clinical and cost effectiveness or value.
Nice. Thank you, Dr. Tice. If you are a clinician who is here not only to learn, but also to get credit for learning, the CME code has been put in the chat for you. You can enter that in. Seeing as we have run out of time, I'm going to ask Dr. Tice for a short, succinct final thought from today.
Yeah, I mean, just as I think Dr. Hodge is trying to highlight, I mean, this is really challenging work because we're forced to make decisions when we really don't have adequate evidence. And part of the reason is because I didn't go into this, but that drug companies are allowed, if something is really impactful, they're allowed to provide less data than we normally would require for FDA approval. In the past, used to be 2 large randomized trials demonstrating efficacy, but they're able to get some breakthrough drug designations and things like that to subvert the usual evidence requirements. And so it ends up being really challenging as a clinician and as a general internist who I'm usually relatively slow to adopt new drugs, and I really want good evidence of efficacy. I feel very uncomfortable with many of the decisions. Sometimes the evidence is clear, like the hepatitis C drugs. There was enough evidence at that time to clearly say, they were having huge impacts in preventing cirrhosis, liver transplants, things like that, and improving quality of life. So sometimes we have adequate evidence at the time, but it's very uncomfortable as a person who trained in evidence-based medicine and really wants super high quality evidence for the evidence that for the outcomes that matter to patients, not just surrogate outcomes. So yes, I feel quite uncomfortable with the evidence review, but I also feel like it's the time when we can have the biggest impact on the pricing, and so it needs to be done, and it needs to be done in a high quality way. And so I'm happy to help participate in that.
Thank you very much for sharing your experiences and your insights with us today. We're really grateful, Dr. Tice.
Yeah, thanks. And
Bye,
nice to see many of you, like Jerry and others who are on. Shui, nice to see you. Thanks for joining.
Thanks
All
again
right.
for your talk. Take care,
Yep.
everybody. We'll see you
All right.
next Wednesday.
Great.
Thank you, Dr. Tice.
You're welcome. Bye.